Welcome to State of the Science. This is our third in a series of 6. And, this is a new way of talking about research in the field of type 1 diabetes. This series is going to be a celebration of the power of debate and discussion that women scientists in type 1 diabetes research bring to the table. We have a global audience of scientists here today, and we thank everyone for joining. I'm really going to briefly introduce the panelists, and the panelists will set the stage for the discussion. Then we're going to ask them to weigh in on their thoughts on these 2 to 3 questions, pressing questions in this arena. Conversation is going to be free-flowing, someone like a cross-idea exchange, a town hall, and a respectful debate. And we're going to have 2 different time points to conduct the polls, and the audience will sort of like weigh in at that time and be able to interact. So a few pro tips for attendees: to submit a question, just place it in the Q&A. You can submit questions throughout the presentation. Participants can upvote your favorite questions at the end of the session. We'll answer questions as time allows. And the event's going to be recorded live on the Sugar Science and also be housed out there for future viewing on our YouTube channel. So in the interest of saving most of our time for discussion, I'm going to do very brief introductions of each of our scientists. And I mean, I sort of say this every time. We could really take up the whole time discussing each of them. They are very valued and integral members in the T1D research field, and you can connect with them directly if you have further questions. But this group particularly has had an incredible footprint in the field. So it's Dr. Bridget Wagner coming from MIT. She's the director of pancreatic cell biology and metabolic disease in the chemical biology and therapeutic science program at the Broad Institute of MIT and Harvard. She's also an institute scientist, and her group is research is focusing on the chemical biology of diabetes with the aim of identifying small molecules capable of increasing pancreatic beta cell number and function, and the ultimate goal of discovering new therapeutic approaches for diabetes. Dr. Susan Bonner-Weir from Harvard and the Joslin in Boston. She's a professor of medicine at Harvard Medical School and holds the Diabetes Research and Wellness Foundation chair at the Joslin Diabetes Center. She was elected as fellow of the American Association for the Advancement of Science, AAAS, in 2012, and she received the 2021 Helmholtz Diabetes Award. She studied the pancreatic beta cell in health and diabetes, focusing on the architecture of the islet and its implications for functions, the in vivo regulation of beta cell mass, and changes of phenotype during the life history of the beta cell. I don't see Dr. Stabler. We'll wait to introduce her. She is supposed to be joining us. She's a professor of integrative life sciences, term professor at the Department of Biomedical Engineering, College of Engineering at the University of Florida. Her work is really about, engineering cell-based tissues for the treatment of type 1 diabetes. Dr. Gopika Nair, she's the CEO of Endocrine Bio and the founder of Endocrine Bio. She's previously an investigator at UCSF, where she invented the breakthrough technology of generating stem cell derived islets in Matthias Hebrock's laboratory. She received her PhD in stem cell biology and endocrinology at University of Wisconsin-Madison, the birthplace of human pluripotent stem cells. And she created several projects creating cellular models, CRISPR-Cas9, gene editing, islet on chip, transplantation, and regenerative medicine. Teresa Mastraaci, she hails from Canada and is an assistant professor at Indiana University, Purdue University of Indianapolis, or IUPUI. And her lab uses zebrafish as a model system that has been instrumental in elucidating the role of hypofuscinated EIF5A in growth and differentiation in the pancreas. And further, Dr. Mastraaci has been involved in multiple collaborative studies and papers which serve to continue illustrating the phenotype of type 1 diabetes. And finally, Dr. Linda DiMeglio, she's at the Indiana University School of Medicine. She works in clinical research with a focus on type 1 diabetes prevention, beta cell preservation, and new technologies and therapeutics. Distinguished panelists, will you set the stage for this discussion? In your scientific opinions, what are the pros and cons of mouse models? And if Dr. Bonner-Weir would go first.
Oh, okay. Well, I think for any of the things, the model you use is dependent on what your question. So I think there are many questions we ask that a mouse model is good because you can easily manipulate the genome and you get a whole body, or you assess it as the whole body. You may be doing a specific in the beta cell. So I think there's some positive things. I think there are negative things in that there's so many things that we have learned. We thought we learned about the beta cell in a mouse, and it doesn't seem to hold up when you go to a human islet. And so I think there's a room for mouse models, but that's only depends on the question you're asking.
Dr. D'Amelio? You want
Yeah, I would echo some of what Susan said just now, so that I was thinking about this topic coming into the conversation, and I was thinking there was people say that rats are not people, but some people are rats, and this is like a mouse version of that. But as a clinical trialist, I like the ability of mouse models to be able to give us answers very quickly compared to human trials, which often take multiple years and are much more expensive and difficult to conduct in many ways. But as was said, sometimes what we've found is we've been disappointed when we've tried to take therapies that had worked even in combination in mouse models and apply them to humans and get results in that way. So as somebody on the clinical side of things, I see strengths and weaknesses.
Yeah, that's interesting. The cytoarchitecture of the islets in the mouse and the human are quite different, correct? So yeah. Oh wait, no,
Is that not true?
No, actually, I
Shaking.
think I'm glad.
So
Susan-
clear.
No, yeah.
No, that's I think one of the falsehoods of that floats around the field.
we've heard it so many times, and I'd love to hear your impressions on that.
Well, but you've got the person who's actually identified where those things are different and the
Thanks,
same.
Teresa.
Yeah, yeah.
In the human pancreas, you have probably half of the islets, which tend to be the smaller ones, actually look like a mouse or rat islet with that mantle core organization. It's the larger islets that you get the sort of composite look. It looks, and depending on how you cut it or you look at it, you are stained for it, it looks very different. But what I see when I look at it is that you have aggregates of the smaller ones together, so that you still have areas that have the mantle core, but you have many other areas that if you cut it across looks like completely random. But I think those are the larger islets. The small islets look just like a mouse or rat.
That's an important point of clarification. I wonder, is there a difference in the distribution across the pancreas that is more or less resembling each other in mouse and human?
I don't think there's really a difference in the large and small across. I mean, in both of human and rodents, you have differences in the islets in that the glucagon and PP rich cells are regionally distributed. There are all sorts of different, there are lots of heterogeneity between islets, but I think we don't know a whole lot more than what you just said, that you have regional hormonal differences, and you have the large and small islets across, and there may be a difference in percentage of large islets in some places than others. I'm not sure that that's really conclusive yet.
It actually kind of highlights why mouse is helpful to us.
Yeah.
Right? Because for the longest time we had this idea of what an islet looked like. And then, Susan's beautiful work looking at human pancreas allowed us to kind of see where things were the same and different. And then there's a lot of developmental studies from mouse that now, because we have these genetic tools, we can also understand relationships between islet cell types, right? Like all the original studies of islet differentiation, where all of these ghrelin positive cells were showing up, right, in the cultures, we now kind of understand where those cells came from and what they become and why they're interesting and intriguing, right? So, and mouse has helped us do all of that. So may not be perfect for everything, but it's definitely helped us to understand what we're seeing in a human setting, and at different ages.
I wanted to add a point to that. There is there's some new data from Alpowers and Brizowa's group that shows that early during. Yes, what Susan said is absolutely right. There are the small islets look just like the mouse islets. Small human islets look like the mouse islets. But what we see is during neonatal, right after birth, most of the human islets look very similar to the mouse setting. And then between 2 and 3 months and 2 years of age, they start aggregating and becoming these bigger, larger clusters. And in the adult, there are larger islets that start to look like what we call as a human cytoarchitecture. So what I would say is that there are similarities, but there must be a reason why the human islets, at least the larger islets, they are organized in a certain way, which of course we think that maybe the alpha cells play a major role in the human condition than in the mouse condition, potentially.
Cool. I'll add one more plug for the mouse. As much as I prefer to work with human islets, the mouse is a great model to test compounds. A lot of the work we do is to identify new compounds that we can use in cell culture. And the last thing we want to do is just let it die in cell culture, right? So we want to advance these ideas. But there are certain aspects of small molecule stability and structural, integrity modifications, things like that, the pharmacokinetics that you really at this point can only study in a whole organism. And so it's really valuable for us for those later stages of evaluation.
And I'll just add to what you're saying there that I would say that the human tissues in those cases are very precious because they're hard to come by. And so if you can test things and work out systems in a mouse model before moving it even to a human in vitro model, and then before moving it into in vivo, I think that makes a lot of sense to use it in that way, especially given what you're saying about the overall similarities in the way the islets function and are organized.
Yeah, that's an excellent point. I mean, the tissues are seemingly very valuable and hard to come by. I wondered, so let's shift gears just for a minute. Which new technologies have the best chance for emulating the human islet phenotype? I mean, mouse has been it seems to me, an incredible starting point, but I know several of you are sort of engaging in other ways of exploring islet biology, and there's islet on a chip, organoids, zebrafish. Some people are looking at Drosophila. I mean, what are your thoughts on these new technologies?
I would say that, respectfully, they're not new, right? And I think Susan's point originally is that, we use the system that's appropriate to answer the question.
Right.
And so my lab uses actually mice primarily as our genetic models, but we have human cells, we have human tissues, and we have zebrafish. And the zebrafish are really used for very, very pointed questions. And we just published a really nice study that kind of shows that on, looking at DFMO, which is a drug that's in clinical trial, and the clinical trial is being run by Dr. Demeglio, actually, and its effect on beta cell regeneration. And you can't really do that in a cell culture model, and you can't do that well in a mouse. So, to us, the fish is the perfect place for that because you can easily drug the water and you can look at cellular changes. So, to answer that question, it's the perfect model, and people have been using it for years.
I guess I just meant sort of from a historical perspective. I mean, maybe you guys can clarify this. It's was the mouse the first sort of model that people were using, and then came zebrafish, or was it all around forever? And what was first?
The rat.
Yeah.
The rat was used extensively for years until people were able to genetically modify the mice. And once you started getting knockout mice or transgenic mice took over, and there's much less work done on rats, though there are some real advantages for using rats for some experiments. Again, they're larger, you can do surgical things on it much more easily. There may be, there's some differences in their physiology, even a little bit from the mouse. But the zebrafish has been around since the 70s or something.
Oh,
Not
yeah.
so much for the islet work, but
No, definitely not for the islet work. I think there was some really beautiful pioneering work that was done for pancreas-driven stuff. Didier Stanier's lab is one that stands out when he was in San Francisco as doing like really beautiful genetic manipulation that gave us a lot of the tools that we still use. But I think there's been pancreas work done in almost every model system, right? Like the fly is definitely being utilized for some pancreas work and some
Yeah,
other.
at Stanford right now.
Yeah, and there's groups at the Joslin, and there's groups like in Boston as well that are using it. So I think every model organism provides its own interesting potential to ask questions. And some of those systems have been around for so long that we have great tools to be able to kind of adapt to the pancreas and understanding these questions.
Yeah. Well, let's see what our audience has to say. This will be interesting. I'm going to allow panelists to vote. Hopefully this will work this time. Yes, launch it. So in your scientific opinion, which model stands the best chance of emulating the human islet phenotype? And the poll is up there. You should be able to see it. Can you see it now, panelists?
Yep.
Okay, great. So it's like islet on a chip, organoids, engrafted human islets, anterior chamber of the eye, kidney capsule, etc., living human slices, AI-driven models, non-human primates, zebrafish, Drosophila. Zebrafish and organoids are getting a lot of attention, and islet on a chip. Oh, this is interesting. Let's see, when I get to 70% or 80%, let's see, people are very thoughtful sometimes, taking a moment to really think it through. Okay, let's go at about this time. It's an anonymous poll, so don't feel like you'll be called out. Okay, we're 70. Here's the poll. Okay, so we can see that it looks pretty much like the engrafted human islets people have thought might be very, have a really good chance of emulating human islet phenotype. What do you think of that, panel?
I certainly think that's true, but one has to realize that there are obviously issues with that model, because you don't get the same organization of the vasculature with the cells. We know that endothelial cells in different tissues are different. They either secrete different things or interact differently. So we're getting, if you're doing it in the kidney capsule or in the anterior eye, you're getting different blood vessels that would grow in. And we don't know enough what causes those differences in, is it something that interaction between the cell that it grows into or that tissue it grows into? We don't know that. But that's a possibility. So maybe they'll change to be more like the endothelial cells, but you still don't tend to get the same density of vasculature as you would in vivo, in native pancreas.
Yeah. No, I also want to add to the point that it doesn't allow throughput. So, transplanted models, be it in the eye or the kidney capsule, there's only so many mice you can transplant and so many conditions you can test. So I would say that is the drawback of that model, but it does definitely give you an in vivo environment, which is not possible in an in vitro cellular dish model. But here I think I should plug in my work, right? So the
Yes.
pluripotent stem cell derived beta cells, be it from human embryonic or human iPS induced pluripotent cells, allows us to do have that access to high throughput and be able to also we can modify the cells through gene editing, change, add the SNPs that we're interested to study, change the background to help us, and also have reporters that you can have to as a readout to study specific aspects of the islet, human islet in vitro.
That's an important point. There is a question here. Do humanized mice offer important new capabilities? And if so, for what questions?
I mean, the short answer is yes, they definitely offer great capabilities to have the human gene of any of the ones of interest, you know, actually in the in vivo setting. And then being able to manipulate and mutate that gene is obviously very, very helpful. And then if you go to the larger systems where they've humanized more than just one gene, but rather, you know, add in human cell systems to the mouse, I mean, it's a spectacular opportunity to learn something.
Yeah.
Yeah, they take a lot of engineering though, and they still, like everything, like there's caveats to all of those models that there's going to be drawbacks to a system that's somehow manipulated, right? So we have to take everything with a grain of salt in terms of what we're going to learn from that system. Yeah, there's a lot of really interesting work that's happening through the NPOD consortium and a couple other groups with the living human donor slices. I think those are really offering some interesting insight into islet architecture and islet cell interactions, given that the tissue slices aren't islets that have been taken out of a person and now put into an in vivo setting, but rather it's islets within their own actual tissue setting, right? The vasculature is intact. There's potentially immune cells that are there. Well, so the vasculature's intact. Yeah, I'll give you that one. And then it's actually human tissue, right? So yes, it's going into an in vivo.
Again, it's if you ask the right questions, it's a fantastic system. I agree, but there are some questions that I know people are trying to answer with that I think are the wrong model for trying to answer, because you don't have you don't have blood flow, you have diffusion across everything. The slices are really only 150 to 200 microns thick, which means you're cutting across the top of lots of islets, since most, you know, the average islet, or what we often call the islet equivalent, is 150 microns. You're cutting across. So it's not ideal. There are a lot of questions that can be answered with it. And
I guess the idea
it's an exciting new tool.
Yeah, the idea that you haven't, you know, completely stripped the islet away from its environment, though, the way we do with islet isolation, and we haven't, you know, manufactured an islet content in using IPS or you know, reaggregated islet cells. I mean, I agree with you completely. There's definitely downsides to it, but I think it's an interesting now new use of human islet or human tissue, right?
How rare is it? How rare are those? You know, these live slices. Is it easy to procure them? Is NPOD?
No, it's quite complicated to do it. It was really developed in Europe with Stefan Speyer and Rupnik. Speyer came over to Miami and really helped them set it up, and NPOD is with NPOD, and NPOD is distributing that. You have to apply for with a project like you do for any NPOD tissue, but there is a, you know, you can apply for getting the slices. I think there are a few other places that have started trying to do it on their own. I haven't seen publications from them yet. I don't know that, but NPOD does provide it to other people. I mean, that's what NPOD does.
Thank God for NPOD. I have a question from Vera Kravitz, coming from Canada. Do you think it's possible to, from Colorado. Do you think it's possible to study innervation effects on beta cell function in human pancreatic slices?
Well, I think the same caveats would come with what Susan said about the vessel, right? Like that you are getting only this, you know, small slice of the pancreas, and so things are going to be cut, and because you don't have the connection to the other organs, right? Like then and the other systems, then you
I
might be
think
able
these
to.
guys are patch clampers.
Yeah, no, well, and then which is it would be great. So I think things that are intact, I mean, you might be able to get some information from them, given that they are living, but it comes with those caveats.
Yeah. So I think that's really interesting. What do people think about the Drosophila? That was, I think there was an interesting talk at the NPOD meeting about the use of the Drosophila. I just wonder, I mean, it's just it's a powerful genetic tool. How similar is its sort of islet or its insulin secretion, you know?
They don't have a pancreas. They
No.
have a
Yeah.
fat body, which is sort of the liver, pancreas, whatever.
Combo.
Yeah, combo. So I don't think the physiology is what you can really test, but you certainly can do the genetics and you know, manipulations and overall, you know, sort of pathways.
Mm.
And there are, I mean, same with the fish. People are attempting to create systems with these alternative lower order models, where you might be able to ask kind of metabolic questions in a way that link to the genetics. So I know like in fish, there are some labs that have, if you feed fish embryos egg yolk or high fat cream, you can kind of create like a fatty liver situation, right? And it allows them to then do higher throughput screens and genetic manipulations in a somewhat disease setting. So you can kind of use Drosophila the same way.
Yeah.
But I agree with Susan, like they don't really have a pancreas. So you are limited a little bit in how you can use them metabolically, but the genetics is beautiful.
Yeah. Here's one more question before we go to our poll. What does the panel think about handling the differences in species differences or ESCs, organoids in glycemic set points for stimulation inhibition of both alpha and beta cells from Vishal-Parekh?
Yeah, I can take that question. So that question is, really relevant. So we do see that there's a set point difference between a human physiology versus mouse physiology for, secretion of both insulin as well as glucagon. One thing very interesting thing that we see is, when we transplant human, either human islets or human iPSC or ESC derived organoids into mouse models, after about 2 to 3 months, the set point of the human cells or the mouse actually changes to come to the human setting. So there you get a hybrid system. So of course it's not perfect because the body is still the of the mouse, but the at least in terms of the secretion of glucagon and insulin, the set point becomes that equivalent to that of a human. So it is a difference, but it also helps us to see, hey, what kind of doses of cells or cell dosage we can transplant and still achieve the same level of set point. So it is something that this field has to tackle, but I guess with larger companies that are going into cell therapy directly into human patients, you don't have that difference.
Yeah, that's pretty interesting that they can kind of evolve into that working, style, I guess you want to call it, or whatever you want to call it.
Yeah. The human system kind of establishes there and then lowers the set point of the mouse model.
Interesting. Okay, let's go to our second poll. And second poll is connecting mouse learnings to human islet mechanisms in type 1 diabetes. So ultimately, how might science merge the mouse model learnings with human islet phenotype mechanisms? And we'll launch it. Oops, sorry. So there's three selections: data-driven creations of new paradigms, artificial intelligence, and these cannot be merged. Mechanisms are too different.
That's a hard one.
It's kind of
I'm gonna
limited.
say I don't know if I like any of these answers.
Yeah, that's what I was gonna say. None of the above, right?
All right, this is
Exactly.
going to be good then. This will be a good discussion debate. But it looks like a lot of people are very interested in this data-driven creation of new paradigms. So I'm going to share the results. It looks like people think that data-driven creation of new paradigms might be the way forward. Okay, let's dive into this. What do you think?
Well, what do you mean by data-driven creation of new paradigms?
Well, I think some of these polls came from you guys. So
Okay.
I think this one might have been from Cherie. I'm not sure. So
I read it as
guessing
kind of
it's like you have to use the data to try to drive, to try to merge the learnings.
I
Yeah.
read it, yeah, as an empirical versus, predictive or AI based approaches. But
Yeah, I also read that has, trying to create a new model based on the data that we obtain from mouse versus human. I mean, in my opinion, I would say that, there are definitely transcriptional differences between the mouse and the human, at least definitely in the transcription factors. One example that Matthias's lab, as well as Roland's lab in Vanderbilt has studied is the MAF-A and MAF-B, and which is different between a mouse and a human, the way that they work in a mouse and a human system. So yes, so to combine those two, there are differences, but there are also a lot of similarities. So a data-driven creation of a new model would be the ideal.
A Venn diagram. Well, so Michael Kalwat says, why not merge both? Why not? Should it happen? No, silence. Crickets.
Well, I think I mean, I'm external to this debate in some ways, but I would think that I think there's a real need for continued communication and collaboration, because the skill sets to do one or the other would be very, very different. I think conversations like this are a good entry point into ways to think about ways to take people who are very siloed in different areas of the science and make sure that they're talking to each other. I think that that's something that we are having to be very creative about right now, particularly in this current era where we're all just literally siloed due to the pandemic. But I think of those, when I think about those two areas, I just think of those very different groups that and the bridging of that's going to take time and collaboration.
Yeah, there is
agree
a,
with
I
agree
just,
with Linda.
yeah, it's a very important point, because I think the disease is so heterogeneous, has a heterogeneity. Plus you've got, like we've been talking about, all these different model systems that are so useful, but they're different. How can it all be brought together? And there is, there's this whole, I think Katerina Volz, I think she came from MIT. She's creating this sort of Parkinson. Her company's called Outcomes Razor. And she's trying to do this for Parkinson's, right? She's trying to layer all these different levels of data and, ways of inquiry and kind of squash it together and have something relevant come out of it. But I mean, that's one approach. Do you see others? I mean, what's the best way to drive collaboration amongst people that are doing different things, different science?
I think a lot of them happen organically, right?
Yeah.
We don't exactly live kind of on islands, right? Any university or any academic institution, you have such a varied amount of science happening around you that
Yeah.
going to get these natural collisions of people that do Drosophila research and diabetes, right? And all of a sudden, therefore, you have this new avenue to study something. I think also like the access to to clinical research and paying attention to what is being put out into the world, like as mouse or fish biologists, right? Like we need to make sure that we listen to people like Linda and the work that's coming out of the human side of things to actually be able to say, are we still researching relevant questions in our animal models, knowing there are new ideas from clinical studies that are out there. I just think I remember back to one of the very first NPOD meetings I ever went to, and there was a massive debate about, is type 1 diabetes an immune disease, or is the beta cell somehow involved? And now I think, like, fast forward 10 years later, I think anybody who says the beta cell is not involved is kind of crazy. But I think there's other things that we probably aren't paying attention to. So should we even look outside of our own diabetes world to say, what can we learn from alopecia research? What can we learn from, RA research? Other immune diseases that might tell us something that we're not thinking of because we're still pancreas or immune system focused, right?
Yeah, I think funding. Sorry, Susan. I
No,
think
go ahead.
funding is also going to be critical here, and finding funders that are willing to take more of a risk, which I think is something I've watched evolve over the last decade or so, where innovation and collaboration is more valued. I think as an academic too, that ability of academic institutions to value collaborative work over individual investigator work is also really important. So incorporating team science into kind of the metrics and the ways that we're being evaluated. With that, I'll turn it over to Susan. Sorry.
Well, I think you've said valid points. I think the one thing I would also say is that we have some resources that, all right, they're not completely new, but they've been broadening. I mean, like NPOD, like we were talking about, not just the meeting, but the access to the tissues, and they are the ones who now have the slices are available. Things like the IIDP, which doing for human islets, and they have gone to having much more sort of studies on the islets you may get. They have now Marcella is doing the sort of analysis of those cells so that you can add that to the information you're getting when you use those islets. And I think that and actually I would also say, now that we're all used to Zoom, I think it's much easier to sort of do collaboration with somebody outside of your own university, where you really can have meetings and interactions much more easily than what it has been in the past.
I think mentoring too also has become easier in Zoom, potentially. I think it's important that people that are looking, especially take new strides in new areas, have careful mentorship from people who are in those areas. And I think that we do try to look at the bright side of the pandemic sometimes, and this ability to still need local guidance, particularly for early career faculty and trainees is really important, but then also the ability to just, like, we're all meeting now and communicating now across time zones.
I want to add one point that, nowadays, if you want to send any sort of mechanism derived paper to a journal, they are going to look for a mouse as well as a human model. So that the times have changed. You can't just publish, which is one of them. So in that way, I think people are forced to now work together, different model systems coming together, and then either validating each other in different systems, or, finding differences between at least in our case, the mouse and a human system. So yeah, the way I see is that the field is moving together, where, like how Michael asked about merging the two, or merging the different systems, is the way to go forward.
Even NIH has some incentives to sort of collaborate as you apply for their grants now, right? So there's some of that going on. Maybe even JDRF will, or maybe I'm not aware that they're already doing that, but that could be another place, because JDRF,
I think in particular, JDRF does a great job of knowing who's out there and trying to match make. But I think, to get to Teresa's point, organic is best. Anytime, and maybe this is maybe a global theme that we've been talking about today, because Susan's talked about it as well. starting with the question. What's the biological question you're asking? You can see it many times with waves of new technologies that emerge, that folks try to just think that, okay, that will solve our problems. Let's just apply it and then we will get the answer. And of course that never happens. So, but I think, again, the organic nature of these collaborations is really the best way to go.
And it is helpful when funding institutions do recognize, that we need to make these collaborations and provide incentives for, fostering those collaborations, or even communicating between two kind of disparate fields within the same study. I'm co-chairing a workshop at the NIH at the end of June that's looking at endocrine exocrine communication, right? And it's bringing together people from the pancreatitis field and the exocrine insufficiency field and CF and, a lot of beta cell biologists. And it's kind of like, it's the first time, hopefully we'll be sitting in the same room, but like it's almost like the first time that all of these people who all study the pancreas are actually going to be talking to each other, right? And learning from each other. And so organically, yes, but it's nice when they're also kind of forced to happen a little bit because of funding institutions.
Yeah, I'll say that the touch points though that I've had, so going like, yes, absolutely, in the same room is really important for that kind of collaboration. I would say that I have sampled in the last year. I mean, like even like this conversation now, the ability to like hop in and sample and listen in different ways, and then think about ways that we can integrate what we're doing. We published some data last year suggesting that more women were attending and chairing and speaking at meetings during the pandemic that were virtual. And I think that it allows, since this is a female focused in some ways panel, like knowing that the ability to do these things kind of hybrid does facilitate some of that ability to kind of get to know people and learn what they're doing and figure out ways we can integrate, I think is also critical. But I do miss like that ability to like have those conversations in other settings. So
yeah, and I think it's unfortunate the trainees don't have it at the moment too, right? I know all of my trainees that started at the university when I did in like August of 2020, none of them have been to an in-person meeting yet. So I want them to be able to make those collaborations and connections with their colleagues and meet people who have, done inspiring research that they've read. So hopefully that'll happen soon.
Yeah, it is I do think that it's great. I mean, I think that there are definitely some places for T1D scientists already, but I do think it is fantastic that you all agreed to just, show up here and talk today. I think that really shows that, people are looking for new ways to communicate. And I, for one, am very interested in showcasing, the incredible work that a lot of women scientists are doing in this field. I think that, the paper that came out from, sort of Jessica Dunn was instrumental in that and putting that forward. That was really surprising. And I spoke to a lot of young trainees. They were also very surprised by this. But I think, from Dr. Bonner-Weir's, perspective, it's not a surprise, that is a truth. It's a paper. I mean, it was written up. So I think that these are opportunities to really kind of like get in front of it and try to kind of create something new. This is a new way of doing things, and we're here talking, and you guys are all like scientists involved, and I appreciate that. Oops, she dropped off.
Oh, we lost Susan.
She's gone. Maybe she'll come back in.
Yeah, and Linda, you were part of that study and that paper. Do you want to talk about like any of how that kind of came together? And
so if I'm not derailing this other conversation, I'm happy to.
I think we've covered the mouse pretty heavily.
We're done with mice for right now.
Because I think it's a really important point, right? Like how we kind of have come to have maybe more acceptance of women in science in our community, and how we can continue to foster that.
Yeah. No, it was that was a effort that I was brought into by Jess Dunn and Mark Atkinson, who had recognized that there was a paucity of women in certain settings they were in. And when they contacted me, I was newly on the editorial board of Diabetes Care and had just figured out that I was the only female physician on that editorial board at the time. That's now changed. So then we went to diabetes care, and we said we would like to publish an editorial, and they said, well, you need data. And so we spent about 2 years then getting data, and we were able to publish that there was a paucity of women receiving awards from these organizations, including EASD, ADA, and that there were just real disparities, which I also had not really seen. I mean, I guess I kind of knew, and it was not something that had occurred to me. And so, and I think it's really sparked some organizational changes, which I think will be helpful. And I'm glad that the ADA had already started the Women in Networking group within the ADA, and they wrote a editorial, and ADA itself wrote an editorial to accompany those papers that were, but I do think that I always look for opportunities, and so that's why I remember most vividly those data around the women's the presence of women at the ADA meetings when they were virtual. And it'll be interesting to see what happens, now that it's a hybrid meeting and we're again required to travel if we're going to present as a speaker. But anyway, there's a lot of work to be done. That could be a whole separate conversation, but I appreciate the interest in that work.
Yeah, I think I do think, this, we tried to formulate this to sort of showcase that there are so many amazing women scientists in this field. And it's pretty obvious that there are, just in these conversations. Somebody from the panel Teresa Quatrin has said we need to make sure that we demonstrate with metrics that indeed women are great mentors, and now that women are more in position of doing so. Oh, yeah. Great mentors now that women are more in a position of doing so. That's a great point, because I do think that when you see what women have accomplished and you have access to them in some capacity, you can really get some great mentorship. And that's what you were talking about earlier, Teresa. The mentorship is so important.
That leads into a question I might have for the panel, maybe, is what do you all think are the best or any metrics for measuring mentoring? How do we determine what makes a good mentor? I mean, we all kind of know that when we see it, but what should we be using?
It's great.
That's a hard question to answer.
I know.
Yeah, because I know
good.
In academic institutions, they say that like the student feedback, for example, is probably one of the worst metrics for determining if a professor is actually good. So I would say, probably student feedback might not necessarily be the best metric for figuring out if your mentor is great. So yeah, that's a stumper. I don't actually.
I mean, I agree. Yeah. It's very difficult.
Leo Ferreira says career progression, he thinks, but it's a hard one.
That's hard.
See that, but on the other hand, sometimes a really good mentor can lead a person into another area, and that might look as, oh, they left the field, and then does that mean you're a bad mentor? No, I mean, not necessarily.
Yeah. No,
so
I agree.
it's
right. If your students end up, or your postdocs end up exactly where they want to be, then that would suggest you've mentored them in the right.
That's very effective mentoring.
Yeah, to get them to where they want to go.
Here's one more comment from Teresa Quatrin. To begin with, allowing exposure and first authorship in papers and presentations, step back and pave the way, get rid of egos.
100%.
Yeah. I think it was Mimo Achille had made, I remember talking to him about this idea of mentorship and, what his philosophy was on it. And he said that he basks in indirect light. And I think it's such a beautiful way of putting it, right? And at some point you step back and you get to be the one that says, yes, this person came from my lab, but look at their accomplishments, right? And when their accomplishments stand on their own, that's you can take it as success, right?
That's so right.
I love that saying.
Yeah.
That's really great.
And I mean, with that in mind, I mean, I think that the new cadre of scientists and women scientists, I think they're really ready for this kind of mentorship, and I think they're ready for to get back into the lab, maybe without a mask on once the whole pandemic sort of dies down. But what else do you think? How do you see this new group? I mean, they've been a little bit impacted by the lack of mentorship and sort of the pandemic stuff. I mean, do you think it's slowed science? Do you think it's just, what are your feelings on the last 2 years and how we're sort of off track here, but how it's impacted these young scientists that are coming into the fray, and how you can best support them?
Yeah, well, I think the one obvious way is that it's impacting the scientists, is that like our bread and butter is being able to talk to people. And while Zoom does give us this opportunity, or like any kind of, online platform gives us an opportunity to reach people we wouldn't otherwise in person, the lack of in-person connection can actually, I don't know, there's nothing that's parallel to it, right? Like in-person connections do lead people down roads. And so I think the current crop of students, and mine are exactly at the top of this. Without the in-person connections, they could be hindered in what kind of opportunities they can then get after they're finished with their degrees. So it's something I'm very cognizant of, and trying to figure out ways to help them make those connections, even though they can't do them maybe in person the last year and a half.
Will it take longer for them to, reach their degree or the end of their postdoc, do you think?
Well, no, because we still have scientific publications that will help move students and postdocs through. So the science, I think, has been able to continue to progress. I think it's just the fulsome nature of connections is not exactly how it was, 2 or 3 years ago. Yeah.
Right.
Okay. Well, I think we really, we tapped on a lot of different, topics here, and I think all important topics, to be honest. But I do, just to cycle back to the mouse model, and, I think we can all agree it's been a very important model to this field. It has provided a lot of historical and current day knowledge to add additions to the knowledge base. And it continues to even, be a very widely used model. And so I guess maybe we should do a show of hands, like, do you think it'll still be around in 10 years? It's still relevant?
Yes.
I think everyone agrees, yes, right? Or
Definitely.
yeah. So, okay, so ultimately the mouse still in the game, despite all this plethora of new tools, and still important piece, despite, some rumblings in what we've heard from the community that, it's no longer relevant, whatever. This cadre feels it is still quite relevant. So thank you all very much for, participating. We're a little bit before the hour. If anyone wants to ask any more questions to the panelists, please do so now. Otherwise, I think we'll probably wind things down and wait till the next one. Our next, our next State of the Science is coming up in a couple of weeks, 2 weeks, on the 24th. And it should be an interesting discussion. You'll see some more promotion about it in social media, and we can't wait to have you join us for our next discussion. So thank you very much, everyone. Again, I appreciate you joining this. And we have a lot of thank yous to you all in the chat. And I hope you have a great rest of your week. And thanks for all you're doing in the field of type 1 diabetes. Very important.
Thank you, Monica.
Thanks, Monica.